In this brief presentation, we'll be looking at the structure of lipids and how cells take advantage of their physical properties. The term lipids refers to a variety of carbon-containing compounds that are primarily composed of carbon and hydrogen, but are all hydrophobic. They do not dissolve in water because they're nonpolar, and water only dissolves polar compounds. There are three types of lipids that we'll be looking at today, starting with fats. Structurally, fats consist of a single glycerol molecule and three fatty acid chains. The chemical reaction that brings together the fatty acid and glycerol is the same dehydration synthesis that we've seen previously. The glycerol molecule has three hydroxyl groups. Each one of those can accept the carboxylic acid group from a fatty acid. The resulting linkage between the fatty acid and glycerol molecule is called an ester linkage. Now, just to refresh your memory, we've now covered peptide linkages in proteins, glycosidic linkages in carbohydrates, and now ester linkages in lipids. The end product of three of these reactions is a fat molecule or triacylglycerol. They're also more commonly abbreviated to triglyceride. Now, one small thing to note is that lipids don't form polymers. Polymers are considered to be long chains of repeating subunits. Lipids form distinct molecules of certain sizes and can't polymerize endlessly. You'll notice that with the molecule that we just looked at and the molecules here on the left, they have no double bonds. As a result, they have as many hydrogen atoms attached to them as possible. We say they are saturated fats because they can't accept any more hydrogen atoms. They are saturated with them. One consequence of them being saturated is that they tend to be linear. If you look at the space-filling model at the bottom here, you can see that the whole chain is linear, even though successive carbon atoms are not in a perfectly straight line. Contrast this linearity of the saturated fats with the unsaturated fats. Unsaturated fats have at least one double bond along the hydrocarbon chain. The result of this double bond is a kink in the fatty acid, which bends as a result. Now, let's consider the difference between saturated and unsaturated fats in terms of how they pack together. Saturated fats, with their long, straight tails, tend to sit relatively close to one another. As a result, there are many intermolecular interactions between the adjacent fatty acid chains. These interactions increase their stability and cause saturated fatty acids to be solid at room temperature. Unsaturated fats, on the other hand, can't be packed so closely together. The kink in the fatty acid prevents this. As a result, there are fewer intermolecular interactions between the fatty acid chains, and such compounds tend to be liquid at room temperature. In general, lipids that we consume are solids at room temperature if they have animal origins, such as fats or fats and meats. On the other hand, plant-based lipids tend to be liquid at room temperature due to their unsaturated fatty acid chains. Some saturated fats have extremely long fatty acid tails, and as a result have particularly high melting points. For example, wax, or like beeswax, has very long saturated fatty acids and is very stiff at room temperature. The next group of lipids are called phospholipids. These are very similar to fats, except that instead of having three fatty acid tails, they only have two. Their molecular structure is based around the same glycerol molecule as fats. However, the third position of the glycerol molecule is taken up by a phosphate group. In contrast to the hydrophobic fats, phospholipids have a very hydrophilic region that we usually refer to as its head. You can see that the phosphate group has a charge, as does the nitrogen on the choline group. These charges allow attraction to the polar water molecules, making the head of phospholipids very hydrophilic. This dual nature of phospholipids, having both hydrophobic and hydrophilic regions, is termed amphipathic. Cells take advantage of the amphipathic nature of phospholipids to form their cell membrane. Phospholipids naturally pack together in what is known as a phospholipid bilayer. Now remember that cells in an organism are filled with fluid, largely water, and they generally are bathed in a fluid as well. As a result, the phospholipids assemble themselves with their hydrophobic head facing the water, either inside or outside the cell, and the hydrophobic tails interact with each other in a hydrophobic region of the membrane. The hydrophobic region of the membrane prevents water and many other substances from passing through the membrane in either direction. As a result, cell membranes incorporate proteins to aid in the transport of materials across the membrane. We'll learn about that in a separate presentation. The last group of lipids are the steroids. Steroids are made up of a four-ring structure. Differences between steroids come from the side groups and the isoprenoid tail. For example, the difference between estrogen and testosterone is a single functional group in the place of the isoprenoid tail here. This molecule is cholesterol, which is used in cell membranes like the one we just looked at. Notice the long tail. Can you see any atoms in it that are particularly electronegative? Cholesterol's tail doesn't have any, and as a result is hydrophobic. Cholesterol is therefore amphipathic, like phospholipids. When they're assembled into a cell membrane, they pack with the phospholipids with their polar region facing the water, either inside or outside the cell, and their hydrophobic tails on the inside of the membrane. Later, we'll see how cholesterol is important for changing the physical characteristics of membranes.